We are going to dive deep into the primary reason why you're healthy, which is essentially the quality of the mitochondria, the way they work, the way they are able to process your fuel and create cellular energy in the form of ATP. So there's two primary ways that they do this. One is the fuel and the modulation of the fuel and the amount of oxygen they're exposed to. So these are two novel therapies. And we're going to talk to a really expert Russian biogerontologist he's been doing this for over 40 years. And I think you're going to get some really good pearls from this, like I did. And I think you're going to enjoy it. It was fascinating for me. And I offer some really interesting novel hypothesis towards the end of this interview.
I was always interested what is the best option application of oxygen treatment to stimulate not specific stress resistance. And from many studies, it became clear, paradoxically, that the most efficient interventions is intermittent hypoxic treatment. For instance, when we have some physical activity, when we stress our muscles, when they are contracted, the circulation is blocked and muscle experience some kind of hypoxia. Then, during relaxation, blood delivery is again open and muscles become again oxygen and nutrients. And this is the universal mechanism which is providing continuous recovery of the mitochondria and other cellular structures. So why not to use this natural mechanism for other purposes? The lymphatic system is very sensitive to the changing blood pressure and blood volume because our skull is not flexible. And for instance, if delivery of blood increases, but simultaneously the release of blood is not released, then we have this difference in pressure and we can feel headache. The lymphatic system is providing this drainage of lymphatic cerebral spinal fluid, and it is very much controlled by intermittent hypoxia. The lymphatic system starts to pump much better during these oscillations, and it clears metabolites from the brain much faster. And therefore, we see very practically instant relief of some headaches during hypoxic treatment, reduction of blood pressure. And this meditative state, it develops during the change from hypoxia to hyperoxia because of the improved drainage function of lymphatic system. This is at least how can I explain what I see with my patients.
Welcome, everyone. Dr. Mercola helping you take control of your health. And today we are joined by Dr. Arkadi Prokopov, who is obviously, as you will tell in a moment, has a very thick Russian accent. And he's from Russia, and it's a country that has been exploring a lot of radical innovations that we have not dove deeply into the United States. And he's going to share some of those insights with him today. And I'm particularly fascinated in understanding his perception of mitochondrial function, because that is, without any question or doubt, one of the single most important strategies you can do to optimize your cellular energy. And it's the core of almost everything that you do to improve your health is mitochondrial function. So anything that addresses that, improves that, is an interest of mine. So that's one of the reasons why I invited you here today. So welcome and thank you for joining us.
First of all, thank you very much for invitation. I must say that I'm your follower since 2001.
Wow, that's 23 years. 23 years.
Exactly. I was working in New York City, and I found out that your broadcasts and your books are very motivating, interesting, and since then I'm following you.
Oh, good. Well, you're a wise man. And hopefully you can share some of your wisdom with me.
I hope.
Yeah, so why don't you first start by giving us a little bit of background for your history, because clearly you've been trained in Russia, and
Yes.
have acquired some significant expertise. I'd like to know if you can share that with us now.
Yes, I graduated from Moscow Medical University in 1980. Yeah. And I worked about 1 year in emergency as emergency doctor, but my interest was always in the biomedical research, specifically research with divers, with professional divers. So I entered my postgraduate studies, and I was doing my dissertation on the topic of improvement of stress resistance in divers. And it was very interesting because all people who were involved, they should first of all do experimentation on themselves. So we first graduated as divers, professional divers, and then conducting a lot of experiments in bottle chamber and also in open water and so on. And that was...
Excuse me for a moment, but wouldn't diving be a simulation of hyperbaric therapy?
Yes, of course. In many aspects, diving is actually, it's a kind of simulation, yeah, of hyperbaric treatment. And moreover, we use the bottle chambers hyperbaric oxygenation for many conditions associated with diving trauma, like bends and barotrauma and so on. So I was working 10 years in this research, but then the situation changed in Russia, and these studies were cut significantly, and I just returned back to medical practice. And what I learned during this studying of physiology of diving and the stress resistance of divers, it helped immensely by application of this knowledge to a lot of various diseases, such as asthma, hypertension, chronic inflammation, and some chronic infections. And I was always interested what is the best option application of oxygen treatment to stimulate non-specific, not specific stress resistance. And from many studies, it became clear, paradoxically, that the most efficient interventions is intermittent hypoxic treatment. So since 1994, I was working in Germany first, in a private practice.
Well, but before we go there, I think it might be wise to explore what is intermittent hypoxia therapy. I know what it is, but I think if you can explain that, and then also the origins of its history. Where did it start from? Was it in Russia?
Yeah, actually, scientific application of intermittent hypoxic treatment started in Russia at the end of 70s.
Okay.
Because paradoxically, it was found that intermittent controlled normobaric hypoxia is much more efficient than continuous hypoxia, such as, for instance, in hyperbaric chambers or in the mountains. This discovery was made by Professor Chizhov. He studied hypoxic treatment in various applications. First of all, as a radioprotective treatment, yeah, because during irradiation, if you reduce partial pressure of oxygen, we see then significant protective effect on healthy tissues. Tumor tissues are not protected because they are already hypoxic, and they don't feel this small decrease of oxygen partial pressure. But for healthy tissues, it's a huge difference. And then he found out that intermittent hypoxia is taking place during embryonic development. So in uterus and even before fertilization, there are significant changes, variation of partial pressure of oxygen, oscillations of oxygen in uterus. And it was not clear what is the purpose, physiological purpose of these oscillations. And now, decades later, we understand that this is a powerful mechanism to control quality of mitochondria. Yeah. And intermittent hypoxia is very often in the nature. For instance, when we have some physical activity, when we stress our muscles, when they are contracted, the circulation is blocked and muscle experience some kind of hypoxia. Then, during relaxation, blood delivery is again open and muscles become again oxygen and nutrients. And this is the universal mechanism which is providing continuous recovery of the mitochondria and other cellular structures. So why not to use this natural mechanism to apply it for other purposes, like enhancement of endurance in athletes, and now this is very well known as altitude training. Thousands and thousands of athletes use altitude training to stimulate recovery, to stimulate functionality, and it's a lot of applications.
So can you tell us how it was implemented initially in the 70s in Russia, and then what the most current applications are now, and basically how it's utilized? I mean, the mechanics of it, what it looks like. So
it looks like, yeah, because in a very simple way, it can be simulated without any devices, just by holding your breath intermittently. For instance, now it's very well method by Wim Hof, who just, recovered a very old variant of pranayama breathing, so-called Nishesha pranayama, so it is called. Everybody knows what is Wim Hof breathing, yeah? But we can do the same without stress of hyperventilation, without reducing carbon dioxide, just providing intermittent flow of oxygen-depleted air into the mask. The patient just using the face mask, and the device delivers intermittently hypoxic air with controlled amount of oxygen and room air, or even hyperoxic air. So now we have the fourth generation of such machines. They are called hypoxicators. They have biofeedback, they are computer controlled, and they allow to perform all kind of treatments, all kind of protocols.
Yeah, I've got one of those units. I've had it for about a year, and it's a cube, maybe 2.5 foot cube, maybe 3, somewhere around that, probably 2.5 feet, 30 inches. And it essentially delivers, as you said, this combination of hypoxic, and the amount of oxygen in the atmosphere and at sea level is close to 20%. And
yeah, 21%. So
yeah,
about.
Yeah, so 21%. So that it modulates it down to therapeutic levels, which seems to start about 14%, and can go down even to 10 or even a little bit lower, but that's in the dangerous levels. And you stay there for a few minutes, maybe even up to 7, 8, or 9, 10 minutes, and then you adjust to that, and you get this hyperoxic air. So in the case of my unit, it goes up to 34% oxygen. And then that's only for a minute or two, and then you cycle back and forth.
Exactly.
And then it's an interesting experience because you can't do much in that environment. So typically what you do, or at least what I've done, is listen to music. I mean, you could listen to podcasts, but I don't think it's a good idea. So you listen to music and you go into a meditation.
Exactly.
Yeah.
Exactly.
And that gives you better benefits. And so I'm intrigued with it. And there's not a lot of research on this. Well, maybe there's a lot of research. There's not a lot written that's available in PubMed, that's for sure. I couldn't find it. and maybe I just didn't know what to search for, but I'm curious as to one of the reasons I invited you is to help me understand how this is working. I know it works, there's no question. My conclusion, this is my thesis, and I see how it integrates with your answer, is that a big part of it is it lowers your CO2 levels. And I understand that your oxygen comes up afterwards, but when you lower it increases your CO2 levels because you're in hypoxic air, so that you don't get respiratory alkalosis, you get respiratory acidosis because your CO2 levels go up. But when your CO2 levels go up, it increases the oxygen circulation to your body, even though you're breathing hypoxic air. That's a normal response. So is that part of the mechanism?
Yes, partially it is. It is, but normally when you are when you're getting into hypoxic environment, your breathing increases, and in altitude, it induces hyperventilation and hypocapnia. When you do this breathing with normobaric air through the mask, or as you did in a cubicle, I must say just that in the cubicle, it's not that efficient as using simply a mask, because the volume which you get in the mask is much more physiologically relevant, and you get much faster oscillations. Yes, the effect of meditation increases. Partially, it is because the play of the capillaries, exactly. In hypoxia, capillaries became wider. In our brain, for instance, hypoxia increases perfusion of the blood up to 40% in hypoxia, just a hypoxic response. And CO2 plays significant role if it's, for instance, many people live continuously in a subclinical hyperventilation. So they have much lower CO2 as it better for the mitochondria.
Yeah, and another term for that would be overbreathing.
Overbreathing, exactly.
Yeah, when you breathe too deeply, typically, not you could breathe faster, but mostly it's the depth of your breathing that causes you to expel too much CO2 when you get to really low CO2 levels, and that is dangerous. That is really dangerous.
Absolutely, especially when it's a chronic overbreathing. And because now people live in a continuous stress, this overbreathing establishes automatically.
It's a habit. It's a psychological habit that is really difficult to overcome. I just interviewed Dr. Peter Litchfield on this, and he has a lot. I don't know if you're familiar with Dr. Litchfield's work, but he's one of the experts in the world at addressing the behavioral overbreathing habits with psychological behavior modification.
Yes, I do agree, but I wouldn't say that this psychological component is very significant, because
he would disagree with that.
Really, I don't. Of course, we should discuss this different opinions and ideas, because as soon as people improve quality of their mitochondria, they stop overbreathing.
Really? So that's been your experience? That's been
absolutely
okay,
because where do we get carbon dioxide? We produce from mitochondria. It's a normal metabolite, and if the mitochondria are not active enough, if they are just lazy for different reasons, they just don't produce enough carbon dioxide. And then hyperventilation during stress even further reduces carbon dioxide, and then we get in this vicious circle. So there are two ways. You can increase amount of carbon dioxide by very simple means, like breathing in a bag, like breathing in just a controlled volume, or even simply we just increase physiological dead space. So you increase volume of the air which is pendling in your trachea. If you use mask and the tube, then you increase this air which is pendling, By this way, you immediately increase partial pressure of CO2. And it reduces many symptoms of overbreathing in minutes.
Yeah, it's kind of like breathing into a paper bag of some sort.
Yeah, but bag. This kind of devices that are called hypercommunicators. And
Okay.
there are hundreds, virtually hundreds of them patented, and they were produced. Now we have in Russia 2 or 3 variants of these hypercommunicators.
Yeah.
But nevertheless, it's just a symptomatic treatment.
Right.
Because as soon as you stop it,
It's gone.
overbreathing again,
Yeah.
again, and you have the same problems. The other way, if you stimulate, if you regenerate your mitochondria, if you make them work more efficiently, economically, it produces much better level of endogenous carbon dioxide. It resets the capnometer, which we have in our brain, we have in our arteries, just kind of, we have thermostat, and we have also capnostat, yeah, because normal partial pressure of carbon dioxide is from 35 to 45, but most people are lower than 35. So if mitochondria are functioning optimally, it automatically levels up the capnostat, and we see a reduction and complete elimination of all problems connected to overbreathing.
That's interesting. So I'm wondering if you've ever, and I want to dive deep into what you're perceiving as an optimal intervention to improve mitochondrial function. I've got some theories or perceptions on that too, and I'd like to discuss with you. But before we go there, have you ever played with or explored the use of exogenous CO2 therapy? So rather than allowing your body to rebreathe and increase the CO2 that way, but actually administering CO2 as a therapeutic intervention?
Yes, of course. Yes, of course. First of all, when you stay in a diving chamber, in hyperbaric chamber, there is automatic control of CO2 level. And there are CO2 scrubbers to keep it an optimal level, yeah? So I was engaged in this research.
And how high did they let the CO2 go in those chambers? What percent? Because normally it's a half of 1%, right?
Yeah, it's
no, it's
0.3, 0.3, so about this 0.5, 0.6.
Yeah.
It's already sensible. People already feel it. If you have this higher level for a long time, for continuous time, it makes actually some problems because it makes a kind of headache. It produces discomfort. So
well, excuse me for a moment, but I thought that a common cause of headaches was vasoconstriction, especially for vascular headaches, and the blood vessels in the brain become really tight and they squeeze in the brain, and that, even though the brain doesn't have sensors, the pressure probably causes some discomfort.
Yeah, I do agree.
And then the CO2, when you increase it, that's a magnificent vasodilator and relaxes those blood vessels, and the headache disappears.
Now we make a step in the very serious problem of lymphatic system in our brain.
Okay.
You know, the lymphatic.
I'm sorry, because of the accent, it's hard to say. Is it lymphatic or glymphatic?
Glymphatic system.
With a G? With a G?
Yes, with G, because we have in our brain, we have very limited drainage of the metabolites. And it was the lymphatic system was discovered, I would say, maybe 5 years ago. It's a system which provides circulation of cerebrospinal fluid. And actually, the removal of metabolites runs through lymphatic system. Yeah. And lymphatic system is very sensitive to the changing pressure of blood pressure and blood volume, because our skull is not flexible. And for instance, if delivery of blood increases, but simultaneously the release of blood is not released, then we have this difference in pressure, and we can feel headache. So, and the lymphatic system is providing this drainage of lymphatic pressure, lymphatic cerebrospinal fluid, and it is very much controlled by intermittent hypoxia. Because that feeling of meditation that you had when you were in this oscillating atmosphere, it is caused by small changes of volume in the brain, arteries, veins, and lymphatic system. Lymphatic system starts to pump much better during these oscillations, and it clears metabolites from the brain much faster. And therefore, we see very practically instant release of some headaches during hypoxic treatment, reduction of blood pressure, and this meditative state it develops during the change from hypoxia to hyperoxia because of the drainage, improved drainage function of lymphatic system. This is at least how can I explain what I see with my patients. And of course, I do it with myself.
Interesting. So maybe we can dive into the recommendations you have to optimize mitochondrial function. But before we do that, I can share what I perceive as a useful strategy, because you are correct. Virtually all the CO2 in the body is produced in the mitochondria, electron transport chain, when you're able, ideally, to metabolize carbohydrates, specifically glucose, which breaks down, glucose and fat both break down to acetyl-CoA, but the process of converting fat to supply the acetyl-CoA to the mitochondria is problematic, largely because of the way the electrons are shuttled around. And you actually produce mitochondrial efficiency by 25 to 50% when you're metabolizing fat. And you do that when you're fasting or even intermittent fasting, or just have a high-fat diet. So that's a potential problem, and you're not going to get maximum CO2 production, because the thing that the fuel that optimizes CO2 production is glucose, no question about it. That is the king of the fuels. And you're going to not only increase CO2 production, you're going to increase metabolic water, structured water, sometimes called deuterium depleted water, otherwise known as, and you will also increase ATP production by 25 to 30%. And you reduce reactive oxygen species, which is another issue to be concerned about with longevity. If you have too much oxidative damage, you got a problem. And you reduce something called reductive stress, which contributes to the oxidative damage. So that was a mouthful. So I'm wondering if you can comment on that and help me understand how that integrates into your view of mitochondrial function.
Well, first of all, I must say that if you don't load your mitochondria continuously, they automatically degrade just from misuse.
Wait, what do you mean by load?
The mitochondria can feel only two interventions, two inputs. What fuel you deliver to them and what amount of oxygen you deliver to mitochondria.
Okay.
Yeah. If there is a continuous flow of fuel, nutrients, and continuous stable level of oxygen, the mitochondria undergo degradation. Because during this ad libitum nutrition and ad libitum oxygen, still oxidative damage in mitochondrial DNA results in a growing population of damaged mutated mitochondria.
Interesting.
Mutated mitochondria have smaller DNA molecule. Yeah, the normal DNA molecule in mitochondria 16,4 kilobase, and it's let's say is like this. But normal metabolism results in a continuous mutation, and it makes the mitochondrial DNA smaller because mutations repaired very insufficiently. Just a piece of mutated circular DNA will be cut off and the ends will be glued together. Next step, in a stable situation, what molecules will reproduce faster? The smaller molecules make their copies a little bit faster than the larger.
Mm-hmm.
Therefore, if everything is stable, normal, yeah, very comfortable, the mutated disadvantageous mitochondrial DNA will dominate.
Mm-hmm.
And we see it with the normal aging process.
Mm-hmm.
We see it in some diseases also when accumulating of mitochondrial mutation, especially in neurodegenerative diseases. Yeah.
Yeah.
So the task is to continuously eliminate or help natural process of elimination of mutated mitochondria. This process is taking place normally. It is mitophagy, yeah,
Mm-hmm.
but mitophagy is also not getting better with growing age, yeah. It's also can be it can be reduced or slowed down with many mitochondrial toxins.
Yeah, sure.
And of course with infection.
Oh,
And therefore,
go ahead.
and therefore, if we just help this process, this natural process of mitochondrial regeneration,
Yeah.
we prevent accelerated decline of mitochondrial quality.
Yeah.
And the best tool for this is intermittent hypoxic training. Because when we introduce intermittent hypoxia to the populations of different in the cell, we have two populations of mitochondria. One original wild type mitochondria, they are okay, they are perfect. But the growing population of mutated mitochondria, they are much more sensitive to oscillations. They don't have enough protective mechanisms. Because you know, mitochondrial DNA protects itself. There is a lot of antioxidative enzymes like superoxide dismutase, catalase, peroxidase. But mutated mitochondria don't have enough of these enzymes, so they are just killed by
I have a
oxygen
question.
oscillations.
Does mitochondrial DNA have histones?
No.
They
As
don't.
far as I know,
That's only nuclear.
It's naked. It lays naked in the stove, in a very hot stove. You know, the latest research shows, yeah,
oxidative stress. That's where most all of the
oxidative
oxidative stress
stress.
occurs. That's
And also very high temperature. It is
Oh,
shown
really?
now that the temperature of functioning mitochondria is about 15 degrees Celsius higher than the
Wow.
environmental
I
temperature.
never heard that before. That's amazing.
Oh, it's publications since 2020.
I'm not denying it. I just never heard of it. That's
Yeah.
amazing.
And that tells a lot about the stress which mitochondria
So
endure.
they truly are furnaces.
Yes, they are
truly
furnaces.
are. Yeah.
Yeah.
Wow.
And
Molecular biological furnaces. I never knew that.
Absolutely. Yeah.
Yeah. So let me go back to you said there's two strategies to optimize mitochondrial function. One is and they both the same process. Essentially, you need to throttle the fuel up and down. Continuous fuel is a disaster. So continuous oxygen the same level, continuous food the same level. So let's go with the food first. So this would support the administration of intermittent fasting, where you're not eating fuel for maybe the majority of the day, maybe 8 hours a day, maybe 10 hours a day, you're eating and the rest you're not eating. Is that a strategy that you perceive as an optimal from the fuel perspective to address this issue?
Absolutely. What I see, and not me, it's a lot of research and experience in the clinical field, that on the fasting state, when your ketone metabolism is much higher, the mitochondrial energy production is more optimal. When the mitochondria are healthy, when we have healthy mitochondrial population. But when the mitochondrial population is a mix of mutated and healthy mitochondria, that can cause problems. Therefore, many people who start, they cannot start fasting, they cannot start intermittent fasting or ketogenic diet because they have their, let's say, 50% mitochondria are dysfunctional.
Oh, okay.
As soon as we repair mitochondria with gradually introduced intermittent fasting, gradually introducing ketones, And in parallel, intermittent hypoxic training, we see immense improvement of energy
Wow.
metabolism. We see improvement of OXPHOS and ATP production, and most interesting, much more economical. So mitochondria in a idling state, they consume much less oxygen as an average human being. On the other hand, at the physical load or functional load, they are much more efficient. Yeah. So we're optimizing mitochondria, we're improving quality of mitochondria.
Okay. So I'm assuming you're doing this in your lab, is that correct?
I do it, let's say, the last 40 years of my life.
Okay, so I'm really intrigued because the big question on the table is how, and I don't know that this is really well done outside the research lab, is where they have like assays like the seahorse assay. How do you assay mitochondrial function? There is no simple test to see how.
Absolutely.
Yeah, so
it's very difficult. It's very challenging on the lab level, because it's really because we see very much discrepancy between clinical results and what we see in the labs. In Germany, where I work, last time I worked in Germany also, we have very good labs for mitochondrial diagnostic, like Armin labs and BOVs, and they have mitochondrial energy test. Yeah, so it shows the mitochondrial quality in you take the just blood and they separate leukocytes, and from leukocytes they extract mitochondria.
Yeah,
and
let me just insert that your pronunciation makes it somewhat hard. It's leukocytes for in typically used, but
leukocytes, white blood cells, white blood cells.
Right.
And we see that really, for instance, by myalgic encephalomyelitis, chronic fatigue syndrome, we see that there is a positive correlation. Yeah, most patients show very low mitochondrial energy production. They show also increased proton leak.
So it's very clear the information you're sharing is valid because you're one of the few researchers that has access to these tools to measure mitochondrial function. I wish to God it was a commercial availability. We could advance medicine so much more quickly because that's the best tool to measure how healthy you are, is you measuring, assaying your mitochondrial function. And there's no good commercial assays for it. There's just don't exist. So
that was good news. But the bad news is that we don't see always a really serious correlation, because we see sometimes that mitochondrial energy analysis shows there is improvement, but we see no clinical improvement. And the reverse also. So we see a drastic clinical improvement, but very, very low or not at all mitochondrial. And it can be explained. It's not something, it can be explained because mitochondrial populations are heterogeneous. They are very different. What we see in the blood, in white blood cells, it comes from bone marrow, and the clinical symptoms we get from neurons, we get from muscles, from liver. And there are different populations of mitochondria, and what is going on in the brain, we don't see immediately in the white blood cells.
Yeah, it makes perfect sense. So most of the mitochondrial assays are using white blood cells to measure?
They are standard lab tests by Armin labs. I can send you just links. I think they have also patients from United Kingdom, from United States. So that's no problem. Therefore, the most relevant mitochondrial tests, to my experience, are very simple clinical tests.
Okay, what are they? Enlighten us.
Basic level of lactate in the blood.
Oh, blood lactate levels.
Of course, blood lactate, because the more lactate the patient has, and it normally correlates with, for instance, with chronic fatigue syndrome, it shows systemic mitochondrial dysfunction.
Correlates
well.
Then
there are functional clinical tests, just physiological tests. For instance, for healthy people, this can be VO2 max.
Mhm.
VO2 max, which can be done very simply, even at home setting, with a stationary bike
Mhm.
or treadmill,
Mhm.
or running 12 minutes, like Cooper test, yeah. And you can correlate it with the VO2 max. The correlation is very high.
No, so let me discuss that because I think the lactate might be better, and I'll tell you why. Because there's I've been was a runner for almost 45 years, over 4 decades. Because I started very early in my life, and I quit like 15 years ago, and I got to be pretty good with respect to endurance training. And I know there's an adaptive phase, and when you first start, you're not that good. It doesn't mean you're not healthy. It's just that you're not good at processing this. And there's a certain level of continuous activity you need to do to get those adaptations. But so that somewhat confuses the interpretation of the data. But whereas blood lactate is more of a metabolic level, your biological level, because when your lactate levels increase, it means you're reverting to anaerobic fermentation or glycolysis in the cytosol to generate lactate. And that usually is correlated with reductive stress because lactate is a reductant. And when you have reductive stress, your electron transport chain does not work that well. It gets really slowed down. So that makes perfect sense that the lactate levels, and you can actually measure the lactate to pyruvate ratio, the NADH to NAD ratio, and there's glutathione reduced to oxidized glutathione ratio, probably CoQ10, the same thing. So those are different ways that you can assess what's going on biologically. But it would seem that the lactate levels they're not related to this training effect that you'd have to do if you were exercising to get that. So I don't dispute that the VO2 max is a useful tool, but I think it needs to be interpreted in the context of all those other variables.
Yes, but there are some details. First of all, a physiological level of lactate is pretty high in healthy people with healthy mitochondria, because lactate, especially for instance, in the night, it's a very important fuel for brain, for neurons, for astrocytes, and also for our heart. Yeah. So there is because lactate has not only as an energy giving fuel, it has also signaling function. Yeah, it works like for so it has many functions, not only as a fuel.
Yeah.
But on the other hand, when you are running,
Yeah.
of course, first you are running. The most economical run is when you're burning fat. Absolutely, all marathon winners, they run on fat. And they save glucose for the last acceleration.
Right, the last sprint.
Then for the sprint, and then they have increased lactate level because they go over anaerobic threshold. So I think these are different mechanisms, and I don't see any contradiction here.
Okay, all right. So I'm assuming that there should be the same modulation, this peaking and trough of the lactate levels as it is with the fuels. And so let's finish up on the fuels first. What is in your experience, extensive experience, what is the optimal time for intermittent fasting? What is the window and I know it takes a while. The average person can't do that. 95% of the population is metabolically inflexible, and they have to adjust to this. A few 5% can do it, but most people can't. So, but if you were healthy enough, what would be the optimal? Is it like you shouldn't eat? I mean, Sachin Panda out of Salk Institute does a lot of work in this, and he states that you should not eat more than 12 hours a day. When you do, that's going to be a problem, and you should go up to maybe 16, maybe 18 hours a day. Then you don't eat. So, what is your experience and recommendations in that field?
Oh, an average recommendation is 8-16.
Okay,
so
16.
8, yeah, 16-8. So 16 hours without food, and during 8 hours you can eat. Yeah. With of course, with more experience, with improvement of metabolic flexibility, which is 100% dependent on mitochondria, you can make it narrow instead of 6 hours. It will just 1 hour a month can be compressed. And I personally, I have maybe 5 to 4 hours meal window. Yeah. And it's a very good synergistically works with hypoxic training.
Oh yeah, so I used to do that too until I encountered Ray Pete's work, and then that butts up against this whole concept that when you don't have glucose to fuel the mitochondria, and maybe you can help me understand this because it may be a non-issue if you have a healthy liver, because your liver is the primary source of the glycogen, which is the polymers of glucose that would supply your body with sugar when you're not eating. And if you have a healthy liver, you probably can go more than a day and still have healthy glucose levels. But once you deplete your glycogen levels, then you have to activate your stress hormones, and you have to activate glycogen, or glucagon, adrenaline, and cortisol. And those stress hormones are pathologic, and it's done continuously at high levels, and it will accelerate premature death. There's no question in my mind. They're bad news. They specifically cortisol, it just sucks out the amino acids, the protein from your tissues, your decreased bone density, decreased muscle mass is bad news. So what have you heard that argument? And if you have, how do you reconcile that?
Yeah, this is a very, very important issue. First of all, gluconeogenesis is an extremely important evolutionary biological mechanism. Because you see, if we take, let's say, a herbivore, some cow or deer, and we measure their glucose level, so it's about 100, about as we have in our blood. But if we take a cat or a lion, and if we measure glucose level, it will also be about 100. Why? They don't eat any carbohydrates. So they get glucose from gluconeogenesis, and gluconeogenesis consumes amino acids. Yeah.
Yeah, that's the cortisol. It's what it does.
Oh no, for gluconeogenesis.
Yeah, that's how you turn it on. I understand.
The amino acids, like in the liver, there is a process of deamination, and the resynthesis of glucose. Normal liver produces about 180 grams of glucose a day,
or more.
In humans.
Yeah. Most people don't know
more.
that.
But you should be healthy enough. You should be metabolically flexible. You should have good mitochondria. Well, then it functions perfectly.
But here's the point. In order to flip that switch and produce those, you have to activate it, and you activate it by releasing these stress hormones, glucagon, adrenaline, and cortisol. And those stress hormones have negative, adverse consequences on your biology if activated chronically. So there's no question you can get really healthy glucose levels by doing that, but you're going to do it at a price. So your body wants the fuel continuously, and it will set it. If your blood sugar drops too low, you are dead. Yes, lactate can fuel your brain. Yes, ketones can fuel your brain, but it needs glucose. Without glucose, you're dead. 100%. You're dead.
I do agree because our erythrocytes, red blood cells, they have no mitochondria. They survive exclusively from glucose. Yeah, that's true. But stress hormones are when they are continuously released, of course, it's a catastrophe.
That's what I'm saying.
Where is a pulsatile release, stress and then relaxation for repair, for recovery, stress and then relaxation. This is like periodicity in training of endurance athletes.
Yeah.
If they're overstressed, they got injuries, they got chronic injuries, and aerobic power will be reduced. But if the properly organized training, they have recovery periods when they replenish all the exhausted hormones. And of course, it's a combination of stress and relaxation. Stress and relaxation. Any continuous stress depletes hormones, exhausts steroid hormones, and induces oxidative stress.
All right, so I think I figured it out. In my attempt to explain the dilemma, at least my perceptive dilemma, I answered the question, and it's related to your liver's ability to store glycogen. If you have sufficient glycogen stores, you can go for 16 hours without eating and not activate your stress response. If you go for a lot longer, specifically, certainly after 2 days or 3 days, 100% of almost everyone over 3 days, no one has that much glucose stores or glycogen stores. Then you're going to activate the only way you stay alive is activate the stress hormone. So I think but here's the caution, the caveat, I think, and I'd like your input on it, is that a third, 40% of the people have liver disease. They have an NAFLD, non-alcoholic fatty liver disease, and that impairs your body's ability to actually produce glycogen or glucose through gluconeogenesis and store it as polymers in your liver. You just can't store as much. So then you have to be more careful. You have to be really careful because fortunately, and usually it's things like linoleic acid, omega-6 fats, and too much estrogen. Those damage the liver big time, and toxins, of course. But if you have a damaged liver, it's not going to work as well.
Yes, of course. Yeah.
Yeah. Well, maybe not. Of course, not. Of course, to a lot of people, they don't get that. That's a big issue because liver disease is pervasive.
Yeah. And I think that this pandemic of liver disease now it's just it's a continuation of metabolic unflexibility.
Yes,
Because
100%.
People,
Right.
in the nature, there is always periods of lavishness and fasting. There is nothing to eat for days, sometimes weeks, and animals survive, and people also survive who sign for very long fasting. You know, there is a very successful cancer survivors who survive just because of prolonged fasting.
Yeah, but I'm not disputing that those exist. I don't think that's the optimal strategy, though. I think that you run into the risk because then when you do that strategy, you are activating stress hormones 100%. There's no way you can go more than 2 days and not activate your stress hormones. It's just biologically impossible. So you have to counter the benefit, and there is a benefit against the downside, and it's a balance, you know. And you do it too much, and it's going to be problematic.
It's always a trade-off, of course. You need just to balance it very finely. You need to monitor symptoms, clinical symptoms, just to listen to your body. That's the success of treatment of very serious chronic diseases.
Yeah. All right. So I think you've helped me understand just the dialogue with someone who's really, really smart about mitochondrial function. The people like you are few and far between. Your level of expertise is pretty extraordinary. So I appreciate the opportunity to speak with someone like you. Help me understand it.
Thank you. Thank you very much. You know, I'm biogerontologist also. Yeah.
Okay,
the first application of intermittent hypoxia as anti-aging intervention. I made presentation in 2008 in Cambridge by Aubrey de Grey, in Sense Foundation. But since then I've seen that it works much more obviously in problematic conditions which are really, really dominating now. Yeah. For instance, Lyme disease, borreliosis. Yeah. I applied this treatment since 2006. Yeah. I now have more than 200 patients with chronic Lyme cured with intermittent hypoxic training, of course, also nutritional component and some necessary.
I don't doubt it. And you could probably add dozens and dozens and dozens of other diseases. No question. And
because it's all about mitochondria.
It's all about mitochondria, 100%. I really appreciate your passion about longevity and biogerontology, as you mentioned. But there's no question intermittent hypoxia training works. It's my speculation that it's working largely as a result of increasing CO2 levels. And there's other benefits. I'm not saying it's the only one. No question there's other benefits. But the increased CO2 level is where the magic is. And the CO2 does it in a few different ways, primarily because it forms this cloud. The CO2 directly attaches to the proteins. Specifically lysine and histidine. And it forms this electric cloud over the protein that protects it. And it actually modulates the expression, the functional expression of that hormone or that protein, which are typically hormones. Most of the hormones are proteins. So you can activate them. You just radically increase the efficiency of the hormones or the proteins and the hormones in your body. And then it also, that's why I asked the question about the histones. The histones are proteins too. And these are the proteins that surround the DNA, not mitochondrial, nuclear DNA, and they modulate the expression. So it's epigenetic control of your body's ability to but produce proteins in your body. Have you heard of akkermansia before? It's been around for 20 years. I don't know if
Yes, of course, akkermansia. Yeah, I gave it to my patients.
Okay, good. So you knew about it. So, and akkermansia works primarily because it increases insulin-like substance, specifically GLP-1, glucagon-like peptide, and it helps modulate control. I mean, it's actually been approved now. It has FDA indication for treating diabetes and by secondary implication, obesity. So if you can, and population, it's normally supposed to be about 10% of your microbiome, but most populations now have like less than 1%. So the key is not to take a magical akkermansia probiotic, and they exist and they do help, but the more effective strategy is to give them the fuel, which would be fibers too, if you don't have a problem. Because one of the things when you have microbial imbalance in your intestine, you have an excess of gram negatives, and the gram negatives are the ones when you have that produce endotoxin, LPA, lipopolysaccharides, and when you have too much of that will destroy mitochondrial function. That and omega-6 are the two primary culprits that destroy mitochondrial function, no question. So the key is to modulate that endotoxin production. And interestingly, akkermansia is a gram-negative bacteria that does not make endotoxin. It doesn't make it. And when you augment the production of akkermansia or akkermansia-like compadres, because there's probably more, that's just the biggest one. But when you increase the production of these, there's a microbiological principle. It's called competitive inhibition. So they grow and they crowd out the other bad guys. So you radically reduce endotoxin when you do that. So it's got a number of different mechanisms. And I just think that the CO2, I mean, it could be, it's not in place of, it could be done in conjunction with something like intermittent hypoxia training. And I think the mechanism would enhance it. And it's just the other hidden value of CO2, but I think it all relates to the CO2. I think it's my belief that CO2 intervention is the single best longevity hack I've ever encountered. It's CO2. So I gave it a long dialogue. I was really appreciate an expert like you, knows the field. I mean, this is your discipline. What do you think about that?
I think that, of course, it's very interesting, extremely interesting. But why not using CO2? Because we have CO2 baths, we have CO2
100%. Yeah.
injections in the skin, hypodermic injections. So it's a known, very well-known option. In USA, by the way, there were also some scientists and practitioners who used it. I know, for instance, there was Laszlo Meduna, who wrote a very thick book about his experience with carbon dioxide inhalations.
Yes, it's different. Not as good as because
it's very extreme. There are many facets of CO2. For instance, as it works as a very important guard of our genome at all. Because
for sure.
The most extensive damage for genome is from peroxynitrite. And CO2 neutralizes peroxynitrite. Yeah?
I did not know that. Wow. Do you have any papers on that?
Oh, yes, there's a lot of publications.
Jeez, show because never knew that.
Continuous, the problem is if there are impulses from free radicals and nitrous oxide and O, that's very important. Yeah, that's killing viruses, bacteria, and this also participates in signaling. But if the both are continuously stable on the higher level, it damages all around. Because peroxynitrite is just exactly, it damages genome. Yeah.
Well, it
So
damages everything.
CO2,
It's massive.
CO2, of course, CO2 is helping to neutralize. Yeah, this so it just supports genome stability, and it automatically means it prevents it slows down the aging process. Yeah.
Yeah, and it also supports the histone. It modifies the proteins in the histones, those amino
Yeah.
acids.
Yeah.
So there's two mechanisms. And for those who don't know, it's the nitric oxide that combines almost instantaneously. I mean, it's almost unmeasurable with superoxide to form peroxynitride.
Exactly.
And the half-life of that is, I think it's 1,000,000 times longer than hydroxyl radical, which is, it's not as potent a reactive oxygen species, but it collectively it is, because it lasts so much longer. It could actually travel outside the cell into other cells.
Yeah. Yeah.
Which is just extraordinary. And it's like something normally hydroxyl radical only travels like the distance of a protein and it's gone. It just doesn't last. So it's a localized damage, typically in the mitochondria. But this peroxynitride is bad news. Oh, it's bad news. That is, I got. Do you have paper? If I just look it up on PubMed, would I find that association? Peroxynitride.
I can send you links, publications.
Okay.
Yeah, I have them.
Pretty good. I mean, because there's, Ray Peat was one of the biologists who brought this knowledge to the forefront, and he used some examples of mammals like the naked mole rat, who's a common example in longevity research, and also bats that live 10, 20, 30 times longer than their other equivalent in
Exactly.
Yeah. So what's the mechanism? And there's a good suggestion that it might be CO2.
CO2 in naked mole rats, absolutely clear. They have hypoxic hypercapnia in their
Yes,
wells.
absolutely.
Yeah, that's clear. It was measured, it was shown.
Yeah.
But there is, of course, some evolutionary mechanisms which are outside of this, because in the bats they live in ventilated caves. There is no hypercapnia there.
Yeah.
But they undergo hypothermia. They're extremely well. They are metabolically flexible till extreme because they can induce hypometabolic state, and of course this state slows down aging process.
Okay. Well, good. This is good. All right. So if someone wanted to explore intermittent hypoxia training, what are your recommendations, and how could they find more about what you're doing?
Oh, they can just go to my website.
Okay.
Go to my website, just or type in Google Arkadi Prokopov. Yeah, and they will find, I have a lot of videos with explanations, and now I'm preparing educational seminars for physicians in English. So it's under development.
Oh, well, good. Well, I want to thank you for all your kind work, and it's been great. I was so looking forward to talking with you.
Thank you for invitation. It was great talk.
Yeah, good. Thanks so much for watching. Remember, hit the like and subscribe button so you can get more videos that can help you and your family take control of your health.